Okay, friends, let's move on to some Yeastly Yearnings during our first fermentation, aka the primary fermentation. Let's now turn to the awesome beast called yeast. Remember, this is part of our further fermentation options, the different tools in the toolkit of a winemaker that they can do or not do, or do in this variation or that variation to affect the finished flavors of their wine. And now we're finally starting to ferment, and it's so simple now, so simple, because you know what the yeast do. They do all the heavy lifting. Chuck some yeast into that juice/must. They eat the sugars, they crank out alcohols, we drink the alcohols, we all party, everybody loves everybody, we're loving it. Well, okay, true, but that is a wildly simplified truth. And there are a hell of a lot more things happening during the fermentation process at the micro level, which affect the product. So let's delve deeper in scale down to their level to discover what these critters are doing and other critters are doing, and how they do indeed do their jobs and how they affect the finished wine, which is what we're always striving for. I'm always just trying to push you to understand and respect these processes, so you better understand and respect the wines and can find flavors in wines that you like even more. Now, first off, in the making of wine, there are 3 stages at which yeast and bacteria do their work, unless we otherwise prevent it, which we want to do sometimes. And the main one is the one we're going to talk mostly about in this lecture, and that is the conversion of sugar to alcohol by yeast. So conversion of sugar to alcohol by yeast. That's the process we're referring to as primary fermentation. The bulk of this work's getting done. The second period that these microbes can do some work is the conversion of malic acid to lactic acid by strains of lactic acid bacteria. And that occurs during something we call secondary fermentation. So we're doing primary. Secondary is obviously later, another fermentation that comes next. During that secondary fermentation, by the way, you can also have yeast eating a little bit more sugar, producing a little bit more alcohol, and producing a little bit more CO2, which we can trap in the bottle. That's how you make sparkling wines like champagne. Again, more on that later. Now, the third period at which microbiological activity can do some things is the conversion of ethanol or ethyl alcohol to acetic acid. That's vinegar by acetic acid bacteria. We've talked about these critters, these acetobacter, which we don't like at all. And they're trying to work all the time. We actually hope to be preventing the acetic acid bacteria work from forming at all times. We want to stop that nonsense at all costs to save our alcohol at all costs. And we'll hold off on this secondary fermentation jazz until a little bit later. Let's just focus on the first fermentation for now, and let's get back to the beast called yeast. Yeast are these tiny single-celled fungi, maybe, that convert sugars into alcohol during winemaking and beer making. All of the alcohol that's pretty much been produced in human history has been by these little single-celled critters. And yes, yeast are different from other types of fungi, maybe so different they get their own classification soon. But yeast are different from other types of fungi in that they maintain unicellular growth throughout their entire existence. So they're born, they eat some sugar, they spit out some CO2 and alcohol and heat, they procreate, and then they die, all in a unicellular system. That's it, that's all. They're particularly hardy little critters too. You can freeze them, you can freeze dry them, you can turn them into a powder and kick them back into some fruit juice and they'll wake back up and go to work. It's really super awesome. Again, it's one of the reasons why these critters are so different that they have been for, I guess, since they created these biological taxonomies, that yeast were put under the fungi division, but I think they're going to end up being their own. So it'll be like animals, plants, bacteria, fungi, and yeast. They're going to be that separate because they are quite unique. Now, the species, and there are tens of thousands, if not hundreds of thousands of different species. Hell, it may be millions, I don't know. But the primary species associated with alcohol production is called Saccharomyces cerevisiae. Aren't you so glad you took that Latin back in high school? It's the main workhorse strain that we want, but there are plenty of others, and there are other yeasts that are commonly found floating around fermentation things are ones like Cloacara apiculata, although I think they may have renamed that one by now, and many other species in the genera of Hansenula, the genera of Candida, and the genera of Brettanomyces. Not all of these are terribly desirable, by the way. We love Saccharomyces, all right, but the other ones, maybe some, maybe definitely not others, because they're introducing different components to our fermentation process. But I'm getting ahead of myself, because there are hundreds and hundreds, if not thousands and thousands of unique different strains of yeast present during a fermentation. Hundreds of sub-strains of Saccharomyces alone. So it's not a monolithic little species. It's kind of like saying Vitis vinifera with grapes. Yeah, that's the species, but you now know that Vitis vinifera subspecies include every single wine grape you've ever had. And think of yeast that way too. Saccharomyces is the one we want, but there's tons of different subspecies that fall underneath of it. Hundreds of them. And they can all affect the resultant flavor and perhaps aroma of the wine. Now, this is important to consider because different yeast strains have different superpowers. They have different abilities. They have different abilities to ferment sugars, different tolerances to different temperatures, different tolerances to alcohol itself, which means that in a single fermentation of one vat of juice, just this one fermentation, that you may have some yeast that are floating around in there and eating sugars and producing alcohol up to about 10% alcohol by volume, and then it's too toxic for them and they die. But don't worry, there's other ones that are floating around in there like, oh, we're fine, and they'll produce alcohol up to 13 or 14%. Then it's too toxic for them and they die. But don't worry, there's other ones that are floating around in there that can go 16 or 70% alcohol until they die. So that's just with the alcohol. What you have here is a real microcosm of diverse activity that we actually don't even fully understand completely yet. I know we like to think that, oh, we got it, and we don't understand what... There's too much going on, and some stuff that we may even see that we don't understand what's happening yet. So, with all of this great diversity of critters, and again, there are perhaps millions, if not billions, of unique substrains of yeast all over the planet. Where are they coming from? Well, we've talked about this. They're everywhere, man. They're floating around everywhere all the time. They're in the air around you right this second, especially around fruit and food. Remember, yeast dig sugars. So they're going to naturally congregate in areas where sugars are happening. And that would be vineyards, but really all kind of food, fruit production in particular. They'll naturally congregate on the grape skins, on the pedicels of the grape, on the stems of the grapes going into it. And again, I pointed out the waxy film on grapes. If you see a white waxy or white hueish tint to any fruit, it's probably yeast. They're just out there. And there may be 50,000, 100,000, a million yeast particles, not unique strains, but a million yeast particles and bacteria stuck to a single cluster of grapes. Million of these little critters. It's crazy. Perhaps up to 150 different unique strains of yeasty shizzle make it into a single bucket of grape juice. That's just nature. That's just natural. It's some mad diversity, by the way. And what this means, especially in wildly fermented beverage, which is what humans have had for most of human history, like the last 10,000 years, wildly fermented, which means that with all of this diversity of microbiological activity, there are perhaps dozens or even hundreds of complex interactions happening within every primary fermentation with different yeast at different times, at different temperatures in different parts of the world. It's kind of really cool if you think about it. But is that diversity is awesome and natural fermentation is great because we wouldn't have drank for 10,000 years without it. But is it desirable for winemakers? Is that how it works in the modern winemaking operation? Well, yeah, you probably know the answer to that. Not so much. With all of those crazy things and crazy critters, most of which we don't even know what they are and we're not sure what they're going to do, that puts in too much unpredictability into our future wine. We don't want that. We want, as a winemaker, to be able to start the fermentation when we want it to start, to have it progress the way we want it to progress, and to finish when it finishes, when we want it to finish. We like predictability, and wild, crazy 150 strains of yeast in your juice ain't going to give you that. So how is it that it's changed in modern winemaking as opposed to most of human history? Well, here's the deal. We mostly just figured out this whole fermentation process really quite recently. We we as humans understood what the process was creating, but we had no clue of how it was doing it. We've only truly understood the role of yeast since this dude Louis Pasteur figured it out just 140 years ago. It's been 140, yeah, say 140. So for all of human history, I should say specifically our fermentation history, 10,000 BC up to 1850 or 1860 AD, brewing and winemaking was pretty haphazard. You'd smash up some grapes, you'd have them in your stone crock pot or a crevice in a rock or what in a clay jug or jar, an amphora, and it would just go to work. Smash stuff in there, you watch it for a couple hours, and it'll start bubbling. And it's quite mystical and fun and magical, actually, because it will start kind of bubbling rapidly. Soon it will and it kicks off heat. Oh, and so you have this beverage in some sort of vessel that has sprung to life. It was just juice, a couple hours ago, and now it's cooking and it's bubbling and it feels hot. It's alive. It's alive, man. And it looks like a boiling pot of stew on a stove, but there's no heat underneath of it. There's no fire. So it is mystical and magical. And then what's it going to produce at the end? Alcohol. More mystery and magic. There's no leap of faith of understanding why we have gods for brewing, gods for wine, gods of the vineyard, almighty Isis, Bacchus, Dionysus, help us out. We love you. This is an interesting, intriguing process that for most of our time on the planet, we have not understood what the hell is going on, but man, we're glad it happens. So this is true for grinding up grain and adding water too, by the way. Grinding up grain, making some bread, oops, added too much water. Oh, it had started fermenting. Hey, that made beer. So anytime a sugary solution was formed, it magically came to life on its own. Fermentation was occurring, and we would all celebrate the majesty and magic of the gods that gave us these great products. Now, people didn't really get what was happening, but they figured out how to tweak the process. And they started doing this by using the same pots or the same vessel, the same clay amphora, or at least keeping some of the residue out of it. So you could, once you made your little batch of wine in your clay amphora, be like, you know what, don't clean it, just leave it over here. Leave it over here till we have juice to put in it. Oh, you know what, we actually need to clean it. My kid got into it and spit in it. So before we clean it, let's scrape off the residue of what was inside the pot. Put the residue over here, and then when we have some more grape juice after we've cleaned out, or we have a new fresh amphora or vase or pot or whatever, put this residue back in with the juice, and magically things would happen again. So they were like, oh, okay, we don't know what this is, but if you keep reusing the same pot or tub or bucket, fermentation will keep happening. So it's got something to do with that stuff that's in there. And they were right, but they couldn't see what it was because we didn't have microscopes yet. It wasn't until you invented the microscope and then this guy Louis Pasteur came along that people started to put two and two together. Well, Louis specifically did. And that's because he was commissioned by the French crown, the French government, to figure out why wine spoiled. Yeah, I know a lot of people think that Pasteur did his original work with milk, but honestly, nobody cares about milk. Wine was huge, big business back in the day, still is in France. And so the idea that you're going to lose a quarter or half of your wine for this year because it's going to spoil before it can get to market, that's a huge economic loss. And so they were investing in researchers and scientists to figure out, hey, how do you stop this process? We don't want this to happen. We don't know what's happening. Can somebody figure it out? And Pasteur was like, yeah, I got you, man. So that was a direct quote. Pasteur, I got this. I got this, Frenchies. So from 1863 to 1875, he figured out a whole slew of things which radically changed our whole understanding of fermentation and how we now make alcohol. In no particular order, Pasteur figured out, one, that microorganisms were responsible for destruction of wine. Looking under a microscope, he's like, oh, I see some things in here, and they're doing some bad stuff. Two, he figured out you could kill those bad microorganisms by boiling them. Hey, what do you call that process where you heat up a beverage or a fluid to high temperatures to kill all the microbiological activity so it won't go bad? Do you know what that's called? It's called pasteurization, because Louis is the one who figured it out. All right. Three, Pasteur identified yeast. Woo! High five, Louis. He's the first one, I guess, who saw them and was like, or one of the first, that saw them and said, hey, there's this other critter here. It's not bacteria. It's something else. Let's call it yeast. He also figured out what their role was in alcohol creation. So yeah, Louis is the one that's like, oh, those are the magical little things. Got it. That's awesome. 4, he identified anaerobic versus aerobic processes of microorganisms. So he was looking at different microbes and saying, hey, these microbes actually can't proliferate or live without oxygen, and these ones over here can live with or without oxygen. So in doing so, we figured out kind of number 5, which is oxygen in large amounts facilitated acetobacter. That's the stuff that spoils wine. So boom, put that in the win column. He figured that out for the French government. He's like, yeah, you got to exclude oxygen out of this stuff because this little critter's in there and it will eat your alcohol and turn it into vinegar. Either exclude oxygen or dose it with sulfur and kill everything, something of that nature. But he also figured out that while large amounts of oxygen assisted that bacteria to kill the wine, small amounts of oxygen actually facilitated wine maturation. It made wine better in small trace amounts. So, I mean, this guy, he did it all. Louis was really le bon, to coin a French phrase, and he really radically changed all aspects of wine production back then, which is still impacting the repercussions of which we are still living with in today's world of how we make wine. So let's get back to the yeast beast now and figure out, okay, what are we going to do in the modern winery to take advantage of all of Louis' knowledge and to produce the best quality wine we can? Well, because I've now pointed out the yeast are everywhere, the bacteria is everywhere, and there's tons of them. We don't know exactly what they do and all these complex interactions. We want to limit the wild ones as much as possible. How do you do that? Sterilization. We've referenced this in the last lecture already. One of the best tools in a grape grower or a winemaker's toolkit is sulfur. And I know sulfur has a bad rap because people don't want to have sulfites in their wine and all that jazz in today's world, but it's a terribly effective tool. It's a chemical that can be mixed in different ways, in a powder form, in a gas form, in a liquid form, and it really is awesome in stymieing or retarding or limiting other microbiological activity that you may not want. So you can make different applications of sulfur in a liquid or a powder form in the vineyard and throw it around the vineyard, and it prevents or stymies the growth of fungus and molds and bacteria. You can use it in barrels when you're cleaning barrels after you've used them one round, or even when you're creating barrels for their first use. You burn a little sulfur in the barrel, kills all the microbiological activity inside the barrel, so the barrel doesn't become the culprit in infecting the wine that's put into it. We have already talked about how you use SO2 during harvest. So sulfur dioxide, it can be made into a salt that's sprinkled over the grapes that you've just harvested while you chill them overnight to make sure that you're limiting micro activity. And of course, the main way is that you, in a gas form, can use sulfur dioxide to pump into the press or even pump straight into your fermentation vessel. Once you have your juice and or your must in the fermentation vessel, you flood the vessel, it kills all microbiological activity, and now you're good to go. What it's doing is it's skewing the ecosystem to what the winemaker wants in order to increase their chances of success in producing the wine that they want. So if you have wild critters floating in and around your fermentation that you don't know what they are and you don't know what they're going to do, then you are putting in elements of chance that you're not going to get what you want. So they want to tweak the ecosystem to get exactly what they want. And so they're going to do this first by wiping out the ecosystem to now have a clean, sterile batch of white grape juice or red grape must that doesn't have any funky wild stuff happening in it. Now, how do you initiate the fermentation then if you've killed all the natural yeast that were already going to work, by the way? They don't wait around. So stop all that. Okay, well, how do we restart it then? Commercial yeast strains. You get to pick the strain of yeast that you want. In fact, not just a single strain, but even like a blend of strains. So there are a handful of major brands that specialize in today's world in creating yeast blends that have been carefully selected to build the specific types of wine, that the winemaker wants, or handle specific winemaking conditions or problems that a winemaker may be having. Same is true for beer, by the way. There are hundreds, nay, there are probably thousands and thousands now of isolated yeast strains cultured under laboratory conditions for purity of breed. So they're in, sterile conditions in a lab, and they're picking out this exact Saccharomyces strain that they want, and they know that they only have this strain and nothing else, purity. But they also have cleanliness, so they know there's no other foreign elements, no bacteria floating around in there. And these things are getting super specific, by the way. You can go online and go search for yeast strains, buying yeast strains. You find catalogs of different yeasts from around the world. And these things are getting super specific in so much that you can buy a strain of Belgian lambic beer yeast that is sourced from a specific village in southern Belgium. I mean, the critter is from there, and it's not found anywhere else on planet Earth except they put it in a laboratory and created it, so now you can order it. You can do the same with wine. They have specific wine yeast strain from, say, northern Sonoma region of Rockpile that's made specifically to do its best work with Syrah grapes. And that, again, we're getting really nuanced here. Not only are we picking out particular yeast strains, but we're saying, oh, and these yeast strains kind of cohabitate with Zinfandel and Syrah grapes, and so they have a symbiotic relationship that they do really good fermenting those particular grapes, and it gets the flavors and aromas out of Syrah that you really like. Because it could be that if you put some random yeast from central China, even if it was cultured and it was a decent wine yeast and you liked it, but maybe it's really good for Pinot Grigio, and if you use that strain of yeast in your Syrah, it would put in flavors that you don't particularly like. Again, very nuanced stuff. So you, as the winemaker, with your sterile juice, are going to order your yeast from a catalog, or you might have produced it locally, more on that in a second, and then you're going to get what's called a yeast starter going. And a yeast starter is, you take a little bit of juice in a bucket, okay, it's just fresh juice that's sterile, and then you put in a bunch of nutrients, like vitamins and minerals and stuff. Then you pour in your dried yeast. Most of it's dried. Again, it's super hardy, awesome little critter. You can bake it, you can dry it out, you can put it on a spaceship, send it to Mars and back, and throw it into this bucket of juice, and it will re-energize, wake up, and go to work. And so what you're doing is putting it in, you're giving it a huge shot of steroids by putting it into this yeast starter bucket that's got tons of sugar and tons of nutrients, and they're getting healthy, and they're awake, and they're forming their colony, and they're reproducing. And so you have tons and tons and tons and tons of yeast cells already ready to go. And now with your starter, you go dump that into your 10,000-gallon fermentation vessel that has your red must in it, ready and waiting to go. And now you're off to the races. So you've got a vat of sugar that's sterile, so it has no competing microorganisms. You got a healthy, hungry colony of specialized yeast, and boom, out of the gate, you got a buffet bonanza begins. It's a feeding frenzy. You think about it as sharks in the water, and someone's throwing in the chum, and this thing just happens, and it happens fast, and it can be even violent. Once yeast fermentation starts, heat's building up again, because that's one of the byproducts, which helps kill unwanted biological activity. And CO2 is also being created as a byproduct, which helps push out all the oxygen, which means the bad bacteria, microbes that need that oxygen can't survive. So I'm telling you this so you understand that even once you initiate this fermentation, you have this huge amount of sugary solution. There's as clean as you keep your winery. You do the best you can, but there's still stuff floating around all the time. So you got to get that fermentation going, and it naturally starts to defend itself. So the creation of heat will actually start to kill off some of the lesser microbes that you don't want in there, and the creation of CO2 is pushing out oxygen, so those bacteria that need oxygen, they can't proliferate, and that specifically includes the acetobacter, that bacteria that will eat your alcohol and turn it into vinegar. So the CO2 is actually a vital component of fermentation because it's saving your alcohol from the acetobacter that's always in there trying to eat it. When all of this is happening, now it's called primary fermentation. We're finally doing it. Now again, this already started much earlier with our white grape juice, and the red grape must may have gone through a few other processes, but at some point whether you chilled out your juice for a while or the must was sitting somewhere and you sterilized it, let it blow off, you've let it chill, whatever, now you're fermenting it, and this is primary fermentation. This is when the major amount of work is being done. This is when most of your alcohol is going to get produced. Now, what are the actual products of the fermentation? Oh, haven't you told us enough about this, Boyer? Well, let's get a little more specific, because this is college. Let's get all sciencey for a second. The actual products in a more mathematical sense is that you have one, for every molecule of grape sugar, we could broaden it to sugar, glucose, fructose, sucrose, but for every molecule of grape sugar, it will convert, the yeast will eat it and convert to 2 molecules of ethanol, alcohol, and 2 molecules of carbon dioxide, plus minuscule amounts of other fractional components and a hell ton of heat. But even that is a gross oversimplification, because this process, I just love talking about this stuff. It's wickedly more complex than we give it credit for and that we even understand. Alcohol fermentation, particularly with wine, involves 12 separate reactions with no fewer than 12 different enzymes, as well as several inorganic ions, which are necessary at this micro level for things to come together and work. And I don't even know what an inorganic ion is, but it's fascinating stuff that there is a little bit more going on than meets the eye or even meets the eye looking through the microscope. The end result of this, theoretically, should be 51.1% ethanol and 48.9% CO2 by weight. We're talking about weight. We're not talking about the purity of the beverage. That's just the chemical equation. One goes in, 51.1 ethanol, 48.9% CO2 by weight on the other side, with heat energy being kicked off as well. In reality, because there's all these complex interactions that we're not even understanding yet, some sugars are getting burned up and converted to other things during this complex molecular dance. So alcohol levels are routinely a bit lower than 50% because stuff gets lost in the sauce, so to speak. And heat, let's get back to the heat. Don't forget the heat. The heat is an important thing to consider for a moment more, because primary fermentation can be impacted in a very big way by heat, and sometimes to its detriment. Meaning that too low a temperature in the must or juice will cause yeast inactivity. And again, that would make sense, I think, to you. If you drop the temperature of your primary fermentation down to 0, it will freeze. And there's no biological activity when it's below freezing, so the yeast stop. So no one would freeze your wine, why would you? But if you get the temperature too low, it can cause them to slow down too much and not procreate as fast, and your fermentation starts to slow down and might get stuck. Conversely, or inversely, a high heat in the fermentation, remember, this is heat being created by the process itself, not by the humans. The high heat can also cause alcohol to evaporate from your fermentation, and you don't want that. So you can't let it get too hot, because you're losing alcohol, the very thing that you're trying to produce here. High heat can also burn off a lot of desired flavors and aromas in more delicate wines. Much more on this later. But fermentation gets too hot, will end up producing a wine that may not be anything like what you wanted, because you've lost too much of the good stuff. Most important though, too high of a heat during primary fermentation can actually kill off some yeast strains. And this can lead to what we call a stuck fermentation. We already talked about different yeast strains can tolerate different levels of alcohol. Different strains can tolerate different levels of heat. But if your fermentation gets too hot and too much of your microbiota gets baked or dies, or gets shocked and is just hanging out, it will stop fermenting. It'll stop working. It's not, again, not procreating cells fast enough. And so everything kind of comes to a stop, and it's called a stuck fermentation, and it's super bad. And it could be that the yeast are still alive, but they've been like, boom, boom, a 1-2. They're in shell shock, and they're going, oh God, I got too hot. I can't work right now. I need to take a break. TV time out. So it could be that there's still yeast in there, and there's still sugar to ferment, but they're not doing their work. And when the yeast stop doing their work, they stop producing CO2. Uh-oh. They also stop producing heat. Uh-oh. So the temperature starts to come down naturally, but also nothing's happening. So oxygen can come back in. Bacteria can come back in. What you have is this big, soupy, sweet slop of mess of juice, and other critters want to eat it. So it's particularly dangerous for winemakers during a stuck fermentation because other things, other microbiota can get in there and take over, and uh-oh, now you've lost a batch to some wild-ass, nasty stuff, possibly. Ultimately, when we're thinking about temperature, and again, we're going to talk about this in the next lecture, most yeast do their best work between 22 and 27 degrees Celsius. That's about 70 to 80 degrees Fahrenheit. Now, I said most yeast do their best work. That's their zone. That's where they want to be. That's when they're really humming. They'll work at lower temperatures. Some will work at higher temperatures. Some will work at lower alcohol levels. Some will work at higher alcohol levels. But that's kind of their zone. And speaking of which, what else could kill yeast besides high heat or super low heat? Why, the alcohol level itself. These critters are ultimately suicidal. They can't last forever in increasingly toxic environments, and they're toxic because they're producing so much alcohol that they make their own environment toxic. So I've already pointed out that, yeah, some yeast can only last to 10% alcohol by volume, some up to 17 or 18% alcohol by volume, but eventually all of them will go. All of them will die. There are some yeast strains that are made specifically to make moonshine, and with moonshine, they don't care about flavors, they don't care about aromas, they don't care about delicate intricacies of the wine. They're just trying to produce as ton of alcohol as possible. So you can dump, buckets of sugar into water and let these yeast go into it, and they've developed specific yeast, I think they're called high gravity yeast, that are built to go up to 22, 23, 24% alcohol by volume. The yeast they're super yeast. They're iron yeast. And they'll just keep going. Even those, though, there is a point at which, no, it's a critter and it can't live. It will pollute its own environment until it kills itself, all right? But in the process, it's produced this wonderful alcohol stuff for us. Okay, how long does it take for these yeasties to do this? How long do the yeasty beasties go at it? Well, it depends on the sugar content of the mixture, of course, of your beverage, of your fluid, I should say, of your juice, because the sugar is their food. How much food do they got to eat? If it's only a little bit of food, they won't work long. If it's a ton of food, they'll keep working until they convert it all to alcohol or until they convert it to enough alcohol that it kills them. Other factors affect the speed at which this process occurs as well. You have the acidity of the fluid, the temperature we've talked about, the work ethic of the individual strain we've chatted about, et cetera, et cetera, et cetera. But mostly, it's what style of wine the winemaker is going for is what affects how long yeast work. Is it a white wine or is it a red wine? Is it a fruit-forward wine or are they trying to make an austere wine? Or are they trying to make a dry wine or a sweet wine? Oh, see how right that second. Just stop with dry versus sweet. Any batch of juice can be made into a completely dry wine. Any batch of juice can be made into a semi-sweet wine. That is entirely up to the winemaker. At any given point, the winemaker makes the decision that I'm trying to produce a certain type of wine. And if I want to have some residual sweetness, if I want to leave some sugar in there, then at some point I'm going to go in and physically stop the fermentation process. I'm going to stop the yeast from working through a variety of chemical or heat ways, right? So even the style of wine in terms of its fruity or its austere, those are decisions that the winemaker is taking into account when he or she decides, let the fermentation go for a day, let it go for 2 days, let it go for 2 months, okay? But discounting all those factors and assuming you just want to make as much alcohol as possible, in general, you have a vigorous and perhaps even violent fermentation for the first 48 hours or so. This critter does not play. So they get in there, I told you, especially after you have a good yeast starter, and they procreate and they eat. It's a feeding frenzy. And again, it's magical and mystical and it looks crazy. But it will simmer down usually after about 48 hours for a more relaxed, slower ferment, which, again, over the course of days, weeks, or months, will more slowly ferment out the rest of the sugars. White wines obviously won't go as long as reds. White wines have less sugar. Red wines typically have more, but again, now we're getting back to what style does the winemaker want to make. But we now have some alcohol. And you know what? Isn't that what this whole section was supposed to be about? So who cares about all the other stuff? All we really wanted was alcohol. That's all we care about when it comes to yeast. They provide no other input into the finished wine, right? Wrong, sir. Wrong. Remember, each yeast strain and other microbes, I keep meaning to put that in, and lots of other microbes, are interacting with different compounds in this solution and are affecting the resultant flavors and possibly the aromas. The flavors added from fermentation from the yeast and microbes are referred to as the secondary flavors. The secondary flavors. Now, don't get confused to talk about a secondary fermentation. That's a different fermentation for a different period, all right? We're still in the primary fermentation. When we say secondary flavors, we're talking about all the flavors and aromas that get put into this eventual wine that aren't coming from the grape. The grape still constitutes like the overwhelming majority of what you taste in the finished wine is what grape you started with. But yeast do play a part of adding some secondary flavors. The work of yeast affect what primary flavors are extracted from the grapes. And that's what's dominant in wine. And that's interesting too, if you think about it, that certain yeast will pull different flavors out of a Syrah grape than it will out of a Pinot Grigio grape. Or that this one yeast strain will pull a certain flavor out of a Syrah grape that a different yeast strain won't. So the yeast can affect things that way at a very micro level. But they can also affect mouthfeel. Different strains of yeast may provide some wines with a more oily, creamy feel, while different strains of yeast would make other wines feel more spicy or sharp. And that, again, that's a mouthfeel thing. We're not even talking about flavors or aromas. But now let me get to what flavors and aromas can we specifically attribute to yeast and their actions. Well, you may have seen some of these things described already in wine descriptions on labels and other things, and maybe some of them, when you read them, you're like, what? That's the dumbest thing I've ever heard of. But dig it. Go ahead and put it in the memory banks. Here are some yeastly flavors of note. When you see these words, or you detect these smells or flavors in the wine, think yeast. Things like creamy, yeasty, cheesy, yogurt, beer, lager beer, sourdough bread, sauerkraut, dill pickle, buttermilk, buttery, meringue even, if you know what meringue is, dried leaves, hay, soy sauce even. If you detect any of these, that's yeast. That's not coming from a grape. And some of these yeast-inspired flavors can get into the wine during the primary fermentation that we're talking about now, but they can also get into the wine during aging, during maturation. Talk more about that later. But yeah, some of these descriptions, soy sauce, what? Sauerkraut? What? You may be like, oh, that's too insane on my brain. I don't associate soy sauce with my fine wine consumption. But let me stress this once again. The grape flavors are still what overwhelmingly predominate a wine. If you get any of these flavors and it's the primary main thing that you're tasting, that's a flawed wine. Something has gone horribly wrong. When I say you might detect things like a cheesy or yeasty or sauerkrauty flavor, I'm saying detect. These are things that may or may not be in the background of the wine, that may or may not be elements of the complex structure of the wine that you detect, that may or may not be on the fringe, on the periphery of your palate or your nose as you're smelling. So you would smell this fruit flavor, that fruit, this dried cherry, this, that, or tobacco, and I get a little hint of kind of a yeasty. That's how it works. So yeast do affect these things in greater or lesser extent depending on the wine style and how much they've gone to work, and a myriad of other factors that, again, we don't even fully understand. The crazier cray-cray, the yeast bacterial action in the fermentation, the crazier cray-cray flavors will end up in the resultant wine. And I'm suggesting that here I'm going to rant for the last few minutes that you may bump into some of these things increasingly, because native yeasts have never really fully disappeared, and I think they're starting to make a bit of a comeback, although that's a strange way to put it too. Namely, in times past, winemakers would pride themselves on the qualities of their cellars for the winemaking experience. And beer crafters, breweries the same way. Yes, you want to keep them clean, but not sanitized clean 100, 500, 1,000 years ago, because it was all the microbes and all the stuff floating around that helped start the fermentation and was part of the process. And it wasn't necessarily that this particular room or this cellar was that great or the winemaking equipment was that great, but it was covered with all these microbes that played including blends of yeast which played an intrinsic part of the wine that was produced. It could be considered almost a trade secret, no less, because if you think about the microflora and microfauna in different parts of the world, the same yeast and bacteria don't exist on one side of town versus the other, much less on one side of Earth versus the other. And there are strains of yeast in Belgium that you can't find in California, that you can't find in New Zealand, and ones in Australia that you can't find in India. These are unique, much like plant and animal life. They are plant and animal life at the micro scale. So my final rant here is something I'm going to call micro terroir. Yeah, right. I just made up a new term. I love it. And micro terroir, you may see rear its head now in the modern world with the proliferation of perhaps more wildly fermented wines. Remember, I have stressed what makes wine so exciting and so awesome, and more awesome than all other agricultural commodities, is their sense of individuality, of their uniqueness. They're unique. Every vintage year is unique. Wines are unique from one region to another. Wines within one region are unique from one side of the region to the other. Wines from a group of vines on this side of the vineyard versus that side of the vineyard are unique in comparison to each other, even within the same vineyard block. So all these things together, the physical geography attributes of the grape growing and the soil and the climate, the cultural practices that go into the grape growing, the cultural practices and history and tradition that go into the wine making. Put all of those things together for each unique part of the planet, and that's what we call terroir, as manifested by the unique attributes of the wine that particular winery or that particular wine region makes. I'm suggesting we're now starting to take that down further in scale to a micro terroir level because of what I've just talking about. These microflora and microfauna are unique, as are the grape growing techniques in central Tuscany. And the microbes are unique to Tuscany, and that goes into perhaps why their wines are unique that they make there. And that you could take the same grapes and the same people and maybe even transplant soil to Australia from Tuscany and try to grow the exact same grapes and make the wine the same way, but the microflora and fauna in Australia is different and it won't taste the same in the finished product. Interesting stuff, right? Now, despite the increasing array of commercial yeast that have become available, many believe that the reliance on commercial yeast removes the individuality of the wine. Again, this micro terroir component. So there are a growing number of winemakers returning to the old traditions of creating their own custom wild yeast fermentations. Now, having said that, they still want to start with sterile juice. Don't get me wrong. Some wineries are going full-on wild fermentation. Let it do whatever it wants. You're seeing them. But it's more that most wineries still want to sterilize their juice so they can control the fermentation. But they're going out into their local areas, their local vineyards or their local valley, and isolating out, finding the yeast that are occurring naturally there and finding the good ones and saying, well, we could send this to a lab, but we could do this ourselves. Let's find the individual Saccharomyces strains here in this valley, figure out the ones that are good and really good or really bad, and then sterilize our juice and then reintroduce local microbiota back into our local grapes and our local wine. That's terribly exciting stuff. So more and more isolating local strains that may do best for their local grapes. It's kind of like a healthy ecosystem and symbiosis type of stuff. Now, not to get all, tree hugger, bunny kissing, liberal professor type. I'm really not kind of the Greenpeace kumbaya singing type of dude. But I think there is something to this. There was a study conducted in Macedonia not long ago on red wines that found that wines made with locally isolated yeast strains produced red wines with higher polyphenol content and higher antioxidant content, as opposed to same wines made in same places with commercial yeast that were strains from someplace else. So higher phenols, higher antioxidants, higher good stuff that we like. Now, that study may just have demonstrated that those local yeasts are just really good. And, of course, it could just be a complete anomaly. But it suggests that native yeast that have grown up with the grapes grown in those areas may be better suited for wine production in their locality. And I believe it does play into what's unique about it. And this isn't even happening with wine. There's a current craze in microbrewing across America now, too, called cool ships. Cool ships in the American craft brewing industry, and that's where you take your wort, and that's the extract that you have after you've ground your grain and boiled it, and you get this sweet syrupy liquid that you're going to ferment into beer. But once you get it all extracted out, while you're waiting for it to cool, you put it in a huge, large, shallow pan, maybe out in the barn, out back, or in your garage, or wherever, and you open up all the doors and windows and just let all the natural crap float in and go to work. Natural fermentations, natural microbes will do it. Now, that can make some pretty funky beers. If you like Flemish beers and sour beers, and they're different, they're unique. So, but people are experimenting with it and trying to figure out, well, how can we use our experimenting with here to isolate out certain strains to then go make our other beers that aren't fully naturally wildly fermented. So it's kind of a fun time. This micro terroir idea, by the way, it's the basis for all unique cheeses in the world. And I don't know if you know this, but cheese is kind of a fermented product. Sausages are, coffee is, cocoa is, sauerkraut is, kimchi is, pickles are. All of these products that have been made for thousands of years, they've been made with local microbiota. Nobody was ordering from a lab during the Dark Ages to make sauerkraut in Bavaria. And cheese is no different than wine. There's unique cheeses all over Europe, not because cow's milk is terribly different from one side of France to the other, but the microbiota that make cheese possible are different. So you have cheddar up here and Munster down here and Gorgonzola down there, and that is a product of localness. So I don't want you to go too far into this. I'm not trying to leave my cheese out in the wind here. So back to the points on wild wines. A small but growing number of producers are using native yeast to produce wines and will often indicate native yeast fermentation right on the bottle. I've seen several of these things, and they're growing in popularity. They do encourage a whole slew of bizarre, fruity, sour, sometimes funky flavors. Remember that whole orange wine production thing I talked about last lecture? Yeah, it's experimental, it's crazy, it's fun, but it can be a little hardcore. So do know what you're getting into when you taste some of these things. And don't really consider it that people are experimenting and doing new things, as if you consider all of human history, they're really returning to more traditional ways of fermenting products. But that's a lot just on yeast. Let's wrap up the bread of yeast here, the crust of the crust, and get on to the next topic, and that is whether wild or commercially concocted, these noble little critters are doing all the aforementioned action during the primary fermentation that we've covered in this lecture, and perhaps a whole bunch more that we don't even understand yet. It is during the primary fermentation that they do their work on the juice/must. But what's holding all the juice/must? Where is the swimming pool that these yeast are floating around and having fun in? It's a container, and there are different containers, of course, and the container counts in terms of how the wine is produced and what it will taste like at the end. So let's get away from the yeast and jump into a vat now and talk about different types of container that fermentation occurs in.